and also as a moderator. I would like to especially tell you that this symposium is due to the special efforts of our program director, Dr. Aaron Gray, at the National Institute on Aging. It is Dr. Gray who organized this symposium, and because of her organization success, that's why we're here. And thank you, Dr. Gray. Yeah. So Alzheimer's, as shown here, Alzheimer's disease multimodal addressing project. Those are projects actually quite systematic, I do think extensive in scope, and they are important part of the NIA's grant portfolios. And the overall goal of this project is to generate high-resolution multimodal analysis and connectomes across species as a platform to discover cellular and circuit-level change underlying aging and disease. This symposium will highlight current AD maps research that builds on the power of BICCN's resource to gain insight into Alzheimer's disease. Our session will start with pre-recorded open remarks and an introduction from the NIA Director, Dr. Rich Hodes, and followed by 4 presentations and a final discussion and a Q&A session. I would like to ask audience to hold a question until the final discussion session. Now, Andrew, would you please play Dr. Hodes' video recording? Thank you.
I am Richard Hodes, Director of the National Institute on Aging. I regret that I can't be there in person, but appreciate this opportunity to open this session. The session is illustrative of the way in which the technologies and data generated by the BRAIN Initiative serve the missions across the National Institutes of Health. In the case of the National Institute on Aging, here the building on the BRAIN Initiative Cell Census Network has played a critical role in formulating the Alzheimer's disease Multimodal Atlasing project. Here, the mission of our institute is served by understanding the way in which cellular composition and networks change with aging and establish vulnerabilities involved in neuropathologies such as Alzheimer's disease. The background provided by BRAIN investigators has already served in active research currently being supported by NIA and will continue along these lines of research. In addition, our National Advisory Council earlier this year approved another concept which will use AAV enhancer technologies to further target the elements which change during aging with Alzheimer's disease. I welcome all of you following today's session to meet in the funders opportunity with Aaron Gray, the NIA program officer involved, and invite you to look further at the ways in which your own interests in brain research can serve the missions of NIA as well as the other institutes across NIH. The specific topics will be addressed in 4 brief sessions, followed by a discussion. The first speaker will be Xiangmin Zhu, who will talk about cell circuit level data from AD, Alzheimer's disease mouse models, followed by Maura Boldrini, who will talk about her work to harmonize her NIA-funded data on human samples with the BRAIN Initiative data sets. And then both Mariano Capito and Lydia Ng will speak on their work on the Seattle Alzheimer's Disease Brain Cell Atlas, the largest human brain atlas currently supported by NIA. So again, this I think is a wonderful opportunity for us to showcase the ways in which research supported by BRAIN has funded and been leveraged to serve, in our case, the Institute's interest in aging and in pathologies that occur with aging. We look forward to more contributions and to conversations during the course of this meeting to foster the common interest that we all have. Thank you, and I look forward to the rest of the meeting.
Well, thank you for Dr. Hodes' very nice open remarks. So in the next, I'm going to tell you about our project and funded by the AD as one of the AD MAPS project. So our project is titled Cell Type Specific Neural Circuits Connectomes in the Mouse Models of Aging and Alzheimer's Disease. Alzheimer's disease is the most common cause of the progressive dementias in older adults, but there's no cures for these terrible conditions. And so recently, clinician and basic research has identified neural circuits dysregulation as an early feature of AD, before other pathological features are measurable. And this is important because this new evidence really provoked the questions: is Alzheimer's disease a circuits disease? And it do suggest a new change of a new conceptual framework for AD research. So we propose neural circuits-focused approaches for Alzheimer's disease research as detailed in our recently published topical review. So if you're interested, you can go to check out a paper and find out more details. And the goal for our project is to construct a comprehensive high-resolution connectomes of specific neurotypes, including molecular and anatomic annotations in critical brain circuits, also defects corresponding with behavior deficits and cell-type-specific gene expression change evoked by aging and Alzheimer's disease. And specifically, we map cell-type-specific input-output brain-wide cortical and subcortical circuits connections of the hippocampal formations in a standardized mouse brain atlas framework using established and the next generation of mouse model of AD. So we have assembled a strong multi-investigator team with strong expertise in neural circuits mapping, next generation AD mouse model development, single cell genomics analysis, and mouse brain common coordinate framework atlas development. So as shown in this slide, and we leverage the strong resources offered by UCI Center for Neural Circuit Mapping, the Model AD Consortium, and the Allen Institute for Brain Science, as well as the Weiju's lab at the UT Southwestern and Iwamukumo's lab at the UCSD. We propose to perform large-scale cell-type specific mapping of hippocampal formation circuits to generate cellular resolution connectome data that combine both molecular and anatomic annotations using multiple AD mouse models. We use 4 AD mouse models for the proposed studies, and which together appear to represent the current best models to explore how AD pathology affects brain circuits. We choose to including 5XAD mouse model because it recapturates many AD-related phenotypes and has an earlier and aggressive AD-like pathology, as shown in this table. And then also we use the human A-beta knock-in mice, and this is Model AD Consortium. It's a new mouse model. It's supposed to model the late onset Alzheimer's disease. And recently, we also, the Model AD also generate a new line mouse model called TREM2-47H, and this basically is GWAS identified mutation, and they've also been using to model the late onset Alzheimer's disease. And the last mouse lines interesting. We actually cross 5XAD with TREM2 mutation mouse model because we like to see how TREM2 mutations impact AD pathology development. And given the Alzheimer's disease has strong features, age progressive features, and so we use this mouse model at 3 different age points, and so that we will be able to look at the age progressive features of brain circuit pathological change. So I also should notice that we have extensive interactions with Model AD Consortium, which is also strongly supported by National Institute of Aging. And as my colleague would like me to tell you that they have been making very good progress, as shown up here. All the mice now are currently available at JAX. And for our project, we've been using the two major new models. This actually, I do think it will have gave us a very good impact. So in the next, I will give you like two really give you some examples about the progress. And first, I'm going to tell about our hippocampus circuit mappings using the Alzheimer's disease models. So we have applied monosynaptic rabies tracing to hippocampal neural circuits mapping studies in AD mouse models to determine how local and global circuits connectivities to hippocampal CL1s excite neurons, how those circuits connectivities to be changed in the APP knock-in mouse models. So this study actually I think is quite important because it's really the first study to apply the new genetic monosynaptic rabies tracing system to an AD mouse models. And we found that the AD mouse models showed age progressive and reduced hippocampal CL1 connectivity. And also we found that the CL1 excited neuron inputs shift with the hippocampal regions in AD mouse model. And particularly the age female AD mouse shows significantly reduced supercrime to CL1 projection. And more recently, we mapped the synapse circuits input to dorsal supercrimes using rabies tracing in the 5X AD mouse model. And we found sex and age dependent changes in connectivities, strengths, and patterns of supercrime presynaptic input from the supercrime subregions and other brain regions in the mouse model compared to the control mice. And we identified the significant sex differences for supercrime inputs. And they also found that in the, particularly for the hippocampal subregion, post-supercrime internal cortex, and all those areas critical for learning and memory. So this paper is just recently published, so you can find out more detail. I should emphasize that because we're using improved monosynaptic rabies tracing approach, and this approach allows us to quantitatively compare, to examine the circuit alterations in AD mouse models compared to control mice. And I want to highlight something here, particularly what I want to point out here is that these data slides really show that there's age, particularly age, the middle age 5X80 is clearly showing the significant connectivity reduce. So because as I told you, rabies will be able to calculate the connectivity strengths. And so we'll be able to clear, you can see there's reduced significant overall connectivities compared to white type mice or young AD mice. And so this is another illustration, but I think it is more clear because you can see that it is for middle age, right? So look at the ports distribution in terms of strengths, they're clearly reduced compared to the other genotype and age groups. And these slides, I think it's important. This is actually very interesting because we all know that Alzheimer's disease is foremost age dependent, and also sex specific difference. And here you can see that. Let's just look at the left column. And clear, you can see that in middle age 5X80 mice, male, females are clearly impacted at the difference in terms of the disease age. So I will, for next example, I will quickly introduce our recent work using the spatial transcriptomic analysis of the Alzheimer's disease pathogenesis in mouse model. So we're using the spatial transcriptomic approach called MOFISH. This approach is developed by Xiaowei Zhuang at Harvard, and I do think it's a very powerful approach. And I show up this video here. You can see the individual transcript. Essentially, you can use high resolution image in individual RNA transcripts, and you will be able to see cell structure, right, and the brain region, and even the different level structures. So this is very powerful because this really allows different conventional transcriptome. It really allows we'll be able to spatial biology, allow to do spatial analysis. And for these studies, we use 300 gene probe, and we will basically produce the spatial resolved molecular atlas, and we use 5xAD control mice. In addition, we also use TREM2 neutral mice and TREM2 cross with 5xAD. So shown here, we have 4 group of mice. And I also show, and in this case, we 4 genotype and the 19 clonal mice, and the yield is pretty good. We got, after QC, we get more than 400,000 of single cells. And this MOFISH really is kind of very exciting. So this is the convention, you get this as molecular cluster cell, and based upon BICCN, you can know the cell type, and you can map up to the spatial location, and there's so you have about 30 cell type and a dozen open regions even across the clonal slices, and the multiplexing is a very rich data sets here. And so this really allow, those data maps really allow us to look at cell type of proportion and as well the spatial region distribution. And the way very exciting is that because MOFISH and the spatial identification plugs enable analysis of the plaque microenvironment. And I show here because for our experiment, we'll be able to identify AD pathology, and those are individual cells. You'll also be able to see the transcripts as well. So we'll be able to look at how the cell type change, how the gene expression changes in proximity of the plaques. And I show up here, just this is. This is also validate. People have seen there's upregulatory microglia activations surrounding the AD pathology. And without the control, so it's nice because now I have 4 genotype, we can do the 4 different pairwise comparisons. And I will not, due to time, I will not be able to get too much details, but I just want to say that microglia and astrocytes shows 5xAD mutation induced transcriptional change, but I show less impact from TREM2 mutation. And so I'm also, so basically this is a summary shows that our MOFISH spatial analysis identify both glial and neuronal transcriptional alteration. And the impact, so this intuition basically indicated the impacting the inflammatory responses, microglia and astrocytes. And also very exciting, we actually found that the BDNA signal tuning in the neuronal cells. And this paper now has been accepted in molecular psychiatry. So in conclusion, I told you that the neurocircuitry studies of brain disorders are increasing in point, and we need to consider neurocircuitry-focused approaches for better understanding and treating Alzheimer's disease. The newly developed technology and conceptual advances present new opportunities for studying cortical plasticities, brain aging, and disorders. And while we generate those standardized comprehensive resources, this work will allow for extensive understanding of the brain circuits susceptible to aging in AD, and will facilitate the development of better early diagnosis and a new treatment strategy for AD. I would like to acknowledge that this work is funded by the AD maps. So this is one of the large U1 project, and I listed the major investigators on these slides. And I do want to thank our program officer, Dr. Alan Gray, and Dr. McAvis. They are wonderful. And lastly, I also would like to thank people in my lab. We are a wonderful group of people in my lab. And also, I would like to thank my large number of collaborators. They're wonderful. And also would like to acknowledge NIH, specific funding from NIH. Thank you. Our next speaker is Dr. Mariano Gabido, who is an assistant investigator at the Allen Institute for Brain Science, and he currently co-leads the data analysis efforts of the Seattle Alzheimer's Disease Atlas. No, please.
Thank you, Shanmin and Erin, for putting together this great session. So today, I'm going to talk about the Seattle Alzheimer's Disease Brain Cell Atlas, CAD for short. And this is an initiative that when it was assembled, it has the aspiration or the goals of trying to map or create a comprehensive understanding and a cellular analysis of the progression of Alzheimer's disease. And if we were, when we created this initiative, if we were successful, we will be able to identify with really precise resolution what are the susceptible cell types to AD pathology, maybe being able to distinguish the progression of Alzheimer's disease from and compare it to regular brain aging. And if we are able to do this, to create or identify the molecular pathways that are perturbed with disease. And if we think about creating a comprehensive cellular analysis of AD, of course, we need to resort to the great cellular taxonomies that are created by the BRAIN Initiative. And CAD in particular is an effort that is joined by the Allen Institute, the University of Washington, and Kaiser Permanente, but we work in really close collaboration with the BICCN human and mammalian brain atlas taxonomies and all the BRAIN Initiative teams creating these really detailed taxonomies. So to be able to understand the progression of AD, the first thing that we needed to do was to recruit a cohort of brain donors that span the full progression of Alzheimer's disease. And so what this resulted in was in the creation of a cohort of 84 donors that if you look at the typical or canonical neuropathological assessment metrics that people that neuropathologists compute, you can see that the participants in the cohort span the entire progression of disease. Our cohort is slightly biased toward older donors and is well balanced in sex. And because it has, some of the participants have comorbidities, our cohort is not just an AD cohort, but also an AD and related dementia, ADRD cohort. So what I'm going to talk today and tell you about is the first installment or the first analysis that we did on this cohort on one particular brain region, the middle temporal gyrus. And in the middle temporal gyrus of this cohort, we collected a multitude of neuropathological omics measurements. So what I'm going, maybe in details, what I'm going to tell you today is that how did we use neuropathology to time the sequence of events that occur as AD progresses, and then using this timing of the events, how did we identify the vulnerable populations or the affected populations to AD? And then I will show you that we were even went further and say, okay, what particular populations, where do they localize, and what molecular programs are affected? So, digital neuropathology. So the lab of Dirk Keen at the University of Washington isolated the MTG from these brain donors and stained it with immunostains targeting like protein pathologies and cellular populations. So then to be able to quantify this, we use machine learning algorithms that take the stains, for example, here I'm showing you amyloid beta plaques, and then create masks that select the plaques, and then we are able to quantify them. So basically creating a quantitative neuropathological description of each brain donor. So we harness this QMP together with a statistical machine, Bayesian statistical models that, just to let you know a little bit, are based on the biological assumption that the progression of AD pathology follows an exponential process. When plaques or tangles are accumulating within a brain region, they are not just aggregating, but maybe multiple seeding events are occurring, or maybe these plaques or tangles are breaking apart. So this idea of multiplying and aggregating can be described with an exponential process in linear time. So if we take together the QMP, the really detailed neuropathological information, together with the algorithms, we are able to order the donors in our cohort from low pathology to high pathology, following this exponential increase in pathology. Here, 6010 is the antibody that targets A beta plaques and 88 phosphorylated tau. So now, we can not only order the donors in the cohort, but assign a score of disease progression. This is a score that lives between the interval 0, 1, and tells you, for example, at 0, you are at the beginning of the disease, you have low plaques, and at 1, you are towards the end, and there is an exponential increase, high number of plaques. So now we can look at all of the cohort, all of the neuropathology, and later on the molecular changes in light of this continuous progression score of pathology, CPS. So this is what I'm showing you here. At the top, I'm recapitulating the graph that I just showed you, but then we can look at all the cellular populations and identify, for example, that at the end of the trajectory, when there are a huge number of plaques and tangles, we have astrogliosis, and also if we look along this disease trajectory, we can recapitulate neuronal declines, which is known, recapitulating what's known in the field. But all of these metrics were used to train the algorithm or to feed the data. These metrics are independent, and they represent, for example, CASI is a score of cognitive proficiency. And we can see that as disease progresses, we are also, with an orthogonal and independent metric, we can see how cognitive decline progresses through the trajectory of disease. Also, I was telling you before about the canonical neuropathological assessments, like BRAC or FAL. That FAL measures like A beta is related to A beta pathology, and BRAC to phosphorylated tau pathology throughout the brain. But here, by looking at one particular brain region, we are able to recapitulate brain-wide processes and even cognitive decline. Not maybe recapitulate, but maybe align all the sequence of events that are happening as disease progresses. Not only, orthogonal metrics, but we can look at our quantitative neuropathological assessment in light of this CPS or this, disease progression score. And maybe something to point out is that we identified that the number of A beta plaques, the size of the A beta plaques start increasing, but then they reach a plateau, kind of telling you that in the MTG across multiple layers, there is a characteristic size of A beta plaques. And each color in these plots represents each layer. So now we can see, for example, the neuronal death that occurs in all cortical layers. But all of these allow us to identify two particular epochs of disease. And one epoch in which early on pathology hasn't taken over. There are depositions of A beta plaques and NFTs, but there are still, we call it in the linear phase because the exponential hasn't taken off. And also cognitive declines hadn't occurred yet. And then a late epoch in disease in which cognitive decline starts taking over, and then the exponential aggregation of pathology is happening across all of the layers. So by using QMP, we were able to organize, again, the sequence of events and identify two critical phases that we call one preclinical and the other one observing cognitive decline. So armed with this continuous progression score, we went and looked at the omics data sets. And the first thing that we needed to do was to, as part of CAD, we are profiling each of the 84 donors in MTG with single nucleus RNA-seq. And what I'm showing you here is each dot is a cell representation. I don't have to go into details at this audience. But on gray in this first plot are all the cells, close to 2 million, collected from the CAD cohort. And then color-coded here are cells originating from the brain reference taxonomy, a brain initiative reference taxonomy on MTG. So the very first level of analysis was to do a label transfer of and annotate each individual cell with their own cell identity. But that not only happens broadly, our cellular taxonomies are hierarchical. So we have multiple levels of definition. And you can see that here we have broad subclasses, but then we go into detail and identify multiple cellular types in non-neuronal cells or neuronal cells. And what I need to tell you is that at the non-neuronal, because the reference cellular taxonomies were built on neurotypical reference donors, if that exists actually, but then along the entire progression of disease, we have disease-associated states that emerge as part of the disease. So for the non-neuronal types, we need to decorate or embellish the taxonomy and annotate each of the new non-neuronal types that exist and are disease-associated states. So the result of this was this creation of this refined brain initiative, CAD, cellular taxonomy that is hierarchical. So here I have two pieces, the continuous progression score that maps progression of disease and a detailed description of each donor of the proportions of cells that exist in the donor. So what I'm showing you here is the result of trying to associate the proportion of cells in each donor with the progression of disease. So basically it's the regression result of a linear mixed effect models in which CPS is our variable of interest and we are controlling for age, sex, etc. And these bars here show you the effect size or the association. And a negative bar will mean that as disease progresses, there is a negative association, so neurons are dying. And when we look in detail, what we can see is that many neurons, many SST neurons are dying as disease progresses or are associated with disease progression, but not all of the SST neurons. And this, you will see later, will play a key role because this is telling us something about the level of resolution that is needed to be able to pinpoint the right cellular changes that are happening with disease. We not only identify SST neurons, but also parvalbumin, layer 2-3 neurons excitatory that were well known, subsets of BIP, and then astrocytes and microglia cells were increasing as disease progresses, a fact that I have shown you before also with the neuropathological information. Just to be able to validate or replicate our results, we performed the same level of analysis in DLPFC on this CAD cohort, and we recapitulate most of the cellular changes. And then one thing that we also did was the following. Now that we have a detailed cellular taxonomy, we brought together all the data from 10 of the most comprehensive cellular studies that were collected in the field. And we reanalyzed all of them and put them in the same coordinate reference or annotate all of the cells with the same level of resolution of the CAD taxonomy. Being able to create, we use this actually to replicate our results in additional orthogonal cohorts, but at the same time, if you pay attention to this, we have created a reference coordinate framework for each for the cellular identity that span most of the studies that are collected in the field that have multiple cells, together collecting almost 8 million cells across MTG and DLPFC. So our continuous progression score allows us not only to look at what are the cellular populations that are dying, but also the timing of the events, as I have been telling you before. And it allows us to identify that SST interneurons are one of the first and continuously lost population along of a disease progression, together with a late loss of layer 2-3 excitatory and parvalbumin neurons. And there is an early increase in astrocytes and microglia population. And maybe, as Xiangmin was also pointing, maybe all of this fact that the critical cellular player in neuronal circuit as SST interneurons is being lost early in disease could be an indication that we need to start rethinking a little bit how we approach AD, because we could think about it as a circuit level disease that disrupt critical players that not only inhibit excitatory populations, but play critical roles in these inhibitory like loops. All right. The last thing I'm going to show you is how did we also, as part of CAD, we also profile the cellular locations of multiple cells. And for profiling the cellular location, we use MERFISH technologies. And here we didn't profile the entire cohort, but a subset of donors that have 28 donors that span the full progression of disease, and then annotate the cells. As you could see, that now the excitatory neurons localize where they should localize at each of the layers of the MTG cortex. And we use this data set to validate our results, because if you look here, we are matching the proportions of SST interneurons collected by 10x or single nucleus technologies and MERFISH dataset, which is a completely orthogonal technology, together with the orthogonal datasets that I have shown you before. And when we look at the proportion of cells in the 28 donors that span this subset of the cohort, we can see the cognitive decline. But then to really harness the power of MERFISH and to identify the spatial locations of cells throughout the cortex, we plot, what I'm showing you here is 3 donors that are sampled across 3 different time intervals across CPS. And what we could see is that the loss of these SST neuronal populations is localized to the upper layers of the cortex, predominantly to layer 2-3. So I don't have this in the presentation, but we have a preprint online in bioRxiv that you can go and check. And something that I'm not showing you is how did we study the molecular changes that are happening in each of the cellular populations along CPS again. But maybe to summarize, what I want to stress this idea that what we were striving was to put together a reference framework, not only on the cellular populations harnessing the brain initiative taxonomies, but also along the progression of disease. And being able, with our this continuous progression score based on neuropathology, to time or order the sequence of events that are happening as disease progresses. And just one more slide as a teaser, I will tell you that what we are doing next is that we are repeating all the set of analysis in multiple brain regions, and really like exploiting or achieving this goal of mapping the progression of AD and the vulnerable types throughout the entire human brain. And this is an integration of multiple areas that we have already collected, and you will see soon the creation of novel cellular taxonomies. And with that, I would like to thank all my colleagues. The CAD team is an enormous team expanding, like the University of Washington, again, Allen, Kaiser Permanente, and the general support of NIA and Erin for this amazing session. Thank you.
Thank you. Our last speaker is Dr. Lydia Ng, who is an Allen Institute investigator. And Lydia is currently also the Senior Director of Architecture for Allen Institute for Brain Science Product. Lydia, please.
Here you go. Thank you very much to Chairman and Erin for organizing this session. My job is then to take the baton from Mariano's talk to how do we make all this rich data that Mariano has shown to be available to our research community. So what we want to do is the ability to make all of this data available, starting with that rich clinical data from the 84 cohort from the 80 data. It's a very special sort of data set. along with its digital neuropathology, seeing the image, seeing the quantification, and also put everything, line everything up according to the continuous pseudo-progression score. On the other side, also integrate that with the single-cell data and the spatial transcriptomic data so that users can then explore the relationship between cell types and AD, and especially along the CPS metric. The heart and soul of our CAD product is the portal. So our CAD portal is the place that we want our data community to go into, and from there they can access different tools for seeing neuropathology, looking at cell types, doing data visualization and mapping. And on the flip side, from our portal, we also want to go to the great collaborator tools and also link out to other community portals. Just like how Mariano was saying that there was this sort of strong handshake between the CAD project and the HMBA, similarly on the data platform side, there is a strong handshake, leveraging and interoperability with our BICCN U24 knowledge base project, in which the single cell, the cell type census data is also coming into integration, and hopefully other brain initiative projects as well. And all of this is built on top of the Allen Brain cell knowledge platform. Okay, so in our portal, we want to make this open community resource for presenting data and quantitative analysis. We're going to integrate the single cell molecular data, the neuropathology, clinical data, to get a deep understanding of what happens to cell types during the progression of Alzheimer's disease. As Mariano said, we have the first installment, and the first installment is looking at a relationship of cell types in the middle temporal gyrus and also in the dorsolateral prefrontal cortex. We are expanding to the multiple regions soon. The data is being collected. After the analysis is done, then we can also push it into the portal. And from there, there's links to multiple tools so that you can then do your own analysis and see the relationships. And importantly, you can also download the data and do your own analysis as well. So the only URL you need to remember is c-ad.org. And when you type in that URL, you will get this page, and this is our portal. And from our portal, there's lots of descriptions of what the data is, and then there's each of these little graphics to show the different components of the different viewers, and then also at the end, how people can access and download the data as well. I'm going to go to this one. So for starting with donor and neuropathology, the donor index gives you access and also ability to filter and look at the range of the demographic, clinical, and cognitive information to the cohort. The ABC Atlas, the Allen Brain Cell Atlas, is our flagship visualization product for looking at single-cell transcriptomic and spatial transcriptomic data. Currently, you can see the integration of the MTG data and the DFC data, and also the MTG spatial transcriptomic data. This is also the same place that we've integrated the whole mouse brain taxonomy and the first draft of the human taxonomy, which John and I showed at the beginning of the meeting. MapMyCell is a sort of user-friendly tool that we've launched last year. So not only can you look at the taxonomy and look at our data, you can also map your data to our taxonomy so that you can see to how your cell type matches, and then also use those annotations for your own research. There's lots of community tools to do additional things. A lot of our data is also available through ZZI's cell by gene, in which you can leverage a lot of the computational tools there. And of course, with the ATAC-seq data, all that can be visualized in the genome browser so that you can explore chromatin accessibility for every subclass, and also for different disease stage as well. And as Mariano says, the important metric to look at this data is through the trajectory of disease progression. So there's an additional explorer in which we can view gene expression changes through these progression. Currently, for the MTG data, we will, as other data become available, they will be added as well. And then there is our gene knowledge card, in which not only is the CAD data available, but also summarize resources for that gene over all our resources. Every data set is currently being used by our community. We have a large number of downloads and views, and then also mappings of map my cells. We do a lot of outreach here. There is a booth or desk just out the door here, and we'll also be SFN. And also, if you look at our portal and through the Allen Brain Map, you can find opportunities, a webinar, that sort of thing, to learn more about the CAD data and our other resources as well. So far we have a lot of data that is available to people through, as well as through our visualization portals. They're also through the SageBio network. We have bulk RNA-seq data, SNP data. All our single cell nucleus data is available there. Through our open data set, we can make available our RNA-seq and single nucleus ATAC-seq data. All of the neuropathology images, if you want the raw images, they are there too, along with its quantification, and also our spatial transcriptomic data and our ATAC-seq data through the genomic browser. Coming up soon, we're working on that now. Some of our specimens are also shared, and currently whole genome sequencing is being done with that, and it will be shared through the NIGADS portal. As Mariano previewed, we have omics data for other regions, including the hippocampus, the head of the caudate, and other cortical areas. All of this data will then be used to do integration with a lot of the reference data and reference taxonomy that we're getting from BICCN through the cell census. There will be more quantitative image through other cortical regions as well, and also the mergefish data in DFC. Okay, I had wanted to do a live demo, but it didn't quite work in this scenario. Again, cad.org or visit us at our desk. So I'm going to do a couple of screenshots, but we're going to start with what can you do with each of these tools. So there's the overview that we already showed you. There is the ABC Atlas, and what can you do there? Our mergefish data is there. With an interactive visualization. From there, you can see the spatial localization of each of these cell types. You can turn different cells on and off depending on which properties you're looking at. And from there, you can see which of these cell types decrease in abundance in high versus low pathology. So the snapshot that you saw from Mariano, you can explore that yourself in different regions. We want to know, you can find out where the gene expression of known genes, how do they differ between cell types, and comparing at different disease states. With Map My Cell, you can then map your own data against the CAD cell types, especially if you have something like a spatial transcriptomic data. When your gene panel is kind of small, then you can then map your cells your cells to our sort of higher resolution 10x, so taxonomical CAD data. With the trajectory viewer, you can see the effect of the gene expression through different stages of the disease. And then again, repeating that this is a very sort of rich clinical data that came with the cohort. You can look at all of this data and investigate each of those neuropathology images. The data set's also available as cell by gene, and in sort of it's been cut. Not only can you view the whole data set, it's been cut up by sort of dissections and region, and you can use a lot of their sort of computational tool there, especially the differential gene search. Okay, now we're going to do some screenshots. This is a screenshot of the ABC atlas. It's particularly is showing the spatial transcriptomic data set. This is in the MTG. What you're showing there looks like images, but these are all the dots or all the cells that has been computationally identified in those MERFISH data sets. So this is showing 24 of the donors. These are grouped by donors, so there's more than one piece of tissue from each donor, and then we basically cut out a rough cortical column from each of them. These are now currently colorized here by class. So here we colorize by class. There are, 30K cells, 300K cells here. You can filter by a certain one. So if you only want to do the comparison between GABAergic and glutaminergic cell, you can turn the non-neuro cells off so that you can get to the cells that you want. While we colorize by cell type, you can colorize by all the clinical metadata as well, and also colorize all the cells by the 500 gene panel as well. Here we have shown, this is a zoom and pan viewer. When you zoom in, you get to basically ability to touch every single cell, and through that you can get what glutaminergic type or different clinical metadata this cell is related to. There is sort of, it's not quite a lasso yet. There is a triangle selection tool. We'll figure out how to do a lasso soon, in which you can sort of put a bounding box around the cells of interest to you, and then you can get a readout of its different metadata in that region of interest. All right, moving to map my cell. I was going to say it was easy as 1, 2, 3, but there's only 2 steps here. So first step is you got to prepare your data. There's instructions, and it's actually fairly flexible. There's instructions of how to prepare your data, because the algorithm is going to look for those columns and row headers in special places. You upload this, so this is just a UI, and then you choose what reference taxonomy you want to map to. Here we selected the CAD. As I said, we also have the whole mouse brain and the draft whole human taxonomy as well. And there is multiple algorithms. We also have a correlation-based algorithm, a hierarchical one. What's highlighted here is a deep generative mapping coming from Mariano and his team. That's based on a sort of a probabilistic algorithm to do inferences between your data set and the taxonomy. Once it's done, it will just tell you where to pick up the data. Our next one is a trajectory viewer. Here, well, I'm going to go back a bit. Here we have just search for this CT, well, our calfepsin D gene, and then we want to see how it changes through disease. So what you do here is first you click on that row, and here what you see is actually a summary of that gene across all the major subclasses. So it is the expression value, and in particular, we're seeing that there's high expression for microglia and astrocytes. More importantly is this panel down here that also not only shows you the change in expression through the progression. So here, of note are the I think it's microglia and astrocyte, which has a sort of an upward trend with respect to the pseudoprogression. And then when you click further in, then you can then zoom into the microglia, and within microglia there are different supertypes, and there's one particular supertype that is upward trending, and for that supertype is considered that there's a sort of a notable abundance change. So you can sort of look at this for every gene in the data set as well. All right. And then there's the donor index, which you can see that you can filter and see all the clinical data. If you click on the little blue button, then you can go and see the neuropathology data. This is a Suman Pan viewer, and then you get a synchronized view of the original data as well as the output of the AI-based quantification. The top one is for the hippocampal area, the bottom one is for the MTG, and these are sort of different stains looking for different properties like plaque and tau. And then this is a zoom in, and there's also annotation to see which subregion you're in. Okay, and the last bit is then, again, you can also see the same data by cell by gene. These are great tools for QC and analysis. This is work done by tens, hundreds of people, thanking everybody from the CAD Consortium at Allen, at UW, at the Kaiser Permanente, and then also thanking our funders at NIA. Right.
Now we have come to our final discussion session. So the floor is open for comments, discussion, and questions.
Hi, everyone. Thank you for the great session. We have a couple of questions from some of our viewers online. So a couple of people have a question for basically the entire panel, whoever wants to answer. With these large data sets and the sophisticated approaches that you're using to analyze them, were there any particular observations that you found that were particularly surprising?
I don't know. You take this one.
It's a little bit vague. Surprising. Maybe you can go.
Yeah. Okay. So I think it's I do as I mentioned in my talk, I do think that the new technology advancement do allow us to gain very good insight into molecular neural mechanisms underlying the brain disorder, Alzheimer's disease. Right? And so from my perspective, I do think it's time. It's actually important to really took the neural circuits approach. Right? So because we know is that Alzheimer's disease, traditional people being focused on pathology, molecular features, but we also know that the Alzheimer's disease shows the disease symptoms, cognitive impairment. What's the intermediate stage? Actually, is neural circuitry. Right? So increasingly, evidence suggests that actually we do need to look at neural circuitry mechanism. So that's, I think, is from my perspective. I do think it's not surprising to me, but I think it's important for us to need to bring the perspective to understand the disease mechanism. So this is my take number one. Take number two, I do think that this high-resolution molecular mapping approach, Murphy's and other approach, really give us a very exciting new findings. In my perspective, normally people, yes, we've seen, microglia, exercise, upregulation in around pathway, but use new technology, by using new mouse models, we also clearly see there's a selective neuronal signal deficits, particularly BDNF and other things. And also, Mariana, in your case, you actually found the SST selective loss, right? These people normally think is inhibiting neurons are harder to
I think that I will reemphasize this idea that what was surprising for us that we were successful in organizing the sequence of events, that we were able to pinpoint individual populations that were the first elements. Like if you think about it, you embark on this trip on collecting these massive data sets, but being able to really pinpoint to the key players that are the first neuronal populations being lost in disease so selectively as superficial layers. A fact that is known in neuropathology, maybe it wasn't, it's not really well known or documented, but when AD progresses in the human brain, there is a banding that neuropathologists observe that is in layer 2, 3, and then really deep layers. So, that correlation between the localized pathology and the particular vulnerabilities. That was, I'm not sure if I will use the word surprising. For us, it's really interesting. So that was one of the...
Maybe, Lydia, would you like to add any further perspective? Lydia said just organize it. But it's also very important, right? Because now related to large scale data sets, how to visualize, right? How to allow the, AD community to access the data is very important.
We just give Lydia headaches, and she just solved in a really cleverly way for all of us to be able. So that, just to mention, the CAD tools that we have developed are the tools that we use regularly, because now we need to go back. Or when we were writing or conceptualizing the paper, having the neuropathological viewer, having the cellular populations being lost on disease, or many of the genes that she was showing, you can see how they progress through the vulnerability. I think that all of those are great tools that I hoped you can use broadly.
Next, please.
Hi, I'm Heather Dawes, UCSF, and thanks so much for these great talks. Question for Mariano, nice to see you again, and can attest to the value of these incredible online resources that you and Lydia and the team have put together, because during the talks I was able to kind of go in and partly answer the question that I had. It looked like you had a fairly decent number of individuals in your cohort that had advanced AD pathology like Braak stage 5 and no dementia. So I'm curious if you've taken a look to see whether this progression of cellular pathology, that chain is in any way different or, unbroken. In some of these patients are very elderly with no dementia. So I'm curious if you have looked at that.
So many thoughts there. In the AD community, there are these research lines on resistance and resilience, like people that develop the pathology but don't have cognitive decline, or that have a lot of pathology and still, I said that, but that are really old and don't develop, no? So, but you point to high Braak stages. And one of the things that along our progression score, we project into one single trajectory, just to be able to map the disease. So we are overlooking in a certain way those variations or the resistance or resilience like donors. We don't have that many. From what I remember, we have 4 or 5. In the scheme of things, we consider it a small sample size. what we really pay attention was that we were able to pull a cognitive phenotype. We have a subset of donors that have pronounced memory decline, like cognitive could be, cognitive decline could be organized in 4 domains: memory, spatial visual, executive, and it escapes me, but but okay, it was, they remain really well performant in the other 3, but memory, and those were the ones associated with what people describe as attacks of chromatin erosion, that also, also groups from MIT. So in those particular subset of donors that might be really have a high level of pathology, at the very end of the trajectory, what we observe is that the molecular machinery shuts down to a point that even the chromatin region shuts down. So to the very end of the disease, like the cells enter into a state that are really vulnerable, the entire molecular machinery. So that was one particular phenotype that we really explore and we showcase in our paper. We didn't, given the small sample size, and like we haven't yet looked at resistance or resilience within our cohort.
Okay, thanks. Look forward to more on that. Appreciate it.
Thank you.
Hi, this question maybe is a little related to what you just talked about. From my perspective, I've never worked with RNA sequencing of disease tissue, but a common step in single cell analysis is to filter low quality cells that may be dying or something like that. So is there any use to systematically analyzing cells that you think are quote unquote low quality in this context? And have you found anything interesting?
Should I go, or do you want to?
Yeah,
I can go. Yeah. So it brings me back conversations with people from the group for like a month or 2 about, because the proper way of saying is that cell quality is collinear with disease progression. Like statistically, you cannot disentangle both events. So by looking at it in detail, you could also associate the percentage of mitochondrial reads, that is a common quality, that also act as a way of filtering cells that lead to the recognition of this donor that I was mentioning, this subset of the cohort that has pronounced memory decline, because most of those cells were not able to pass the quality control. So then you could think, if you are used to the single nucleus RNA-seq analysis in this UMAPs representation, that there will be a smear that you can still recognize, annotate, and analyze, but at certain point, neuronal identity is lost, and you cannot, recover those cells. So using that particular, or paying attention to that fact, was that we identify. And in those subset of donors, there was no way of bringing back those cells. The batch effect of the number of reads per cell didn't allow it. So the analysis that we can do is like at the number of cells per donor. How many cells pass the QC? On the other, but then we could think of those subset of cells containing donors that are highly affected by pathology and are at the end of the trajectory. But early on, like pinpointing SST, the molecular changes that are happening in the populations that are dying and the ones that are not, because Lydia was showing you in the CAD transcriptional viewer that you can see the changes and color code, this population is dying, this not, but you still have changes. So all of that early level of analysis can still be performed, and it's like, at least to me, one of the one that could be most fruitful, because there are other members of the team that are working on gene therapies, and they are looking at the earliest changes, knowing the entire disease progression. We are using like AB derived cell type specific expression to try to drive therapy. So
maybe I would like to add a comment, because we're talking cell types, but sometimes particular disease might also relate to cell states, right? So I think the comment is the way. Actually, I do think this is a question, still, ongoing discussion in the field, right? Particularly for the disease situation, when we have to make a threshold.
I mean, in his question in particular was regarding QC, no? And I think that I gave you the full overview and many. Regarding cell state, yes. So I think we are at the point that our algorithms are able to capture not only cell type variability, but certain cell state variability that you can annotate them.
Right.
And I'm not sure if you pay attention, but we call our cell types super types. Not because we are super, but because we had a procedure that we subset the donors and we try to, let's say you create, you over cluster and create really tiny descriptions of the cells. And then you aggregate them until you can see really clear differences. Okay, that's a first level of analysis. But then we subsample our cohort and say, okay, can we identify the same cell types with this subsample, like multiple bags? And then if we are able to identify and create a robust cellular annotation, we are happy at that point. And that came from a necessity, not targeting your question of cell state or versus cell type, but the idea of now we have to deal not only with the, the reference taxonomies, but the taxonomies along disease states.
Okay.
So, the taxonomy has to be robust. Okay. So, anyway.
Thank you. Next, please.
Yeah, I saw that you looked at a lot of cortical samples. The somatostatin neurons that were depleted in the early stages, what subset of somatostatin neurons were those, and what part of the cortex were those in? Was it very broad, or was it restricted to, for example, entorhinal cortex?
No. First, in the in this level of analysis, we focus on MTG and DLPFC, and these were non-Martinotis.
They were the non-Martinotti cells. So
localizing preferentially to the superficial layers.
HPSC expressing non-Martinotti?
I don't remember.
I see. Okay, thank you.
But you can go and ask Lydia to walk you through our tools.
But presumably it's very easy to go through the atlas, the portal, and see whether they're depleted.
Then you have to walk her.
Oh, well, yeah. but you just haven't done it yet.
No, yes, I cannot recall it right now. But
Ed, your comments?
I have a question for Lydia. I think there's a lot of power to mapping against the Brain Initiative reference and the knowledge that's starting to aggregate around there. We mostly heard about molecular and spatial, but could you tell us a little more about what's coming, the sort of aggregate knowledge that will be available?
Yes, so this is why it was important that this data came within the framework of the knowledge base and also in our platform, because this is the space where we want to integrate everything. So the dimension of integration is like by CCF or by anatomic, by taxonomies, and also by genes. So once we have everything aggregated, if the CAD data comes along with the whole brain data, and then we'll have single cell morphologies from our surgical tissues. In the mouse, we can have whole brain morphologies as well, and also physiology. And especially exciting is the integration with all of BICCN, because starting with BICCN, we have a rich mouse data set there. And within BICCN, especially through the HMBA, there's going to be a rich spatial transcriptomic throughout the whole brain for human, marmoset, and macaque. And there's also many groups that are doing developmental version of that. And there's also a developmental mouse. So all of this data coming to some common place where we can jump from one piece of data to another, it's going to be something that's game-changing, and we're trying to figure out how do we integrate it so that it can scale as well.
Thank you, Lydia. So I think Dr. Maura Baldwin is on Zoom. I think you could have we have about a minute for you if you want to make some comment.
Um, yeah, I think, I'm really looking forward to integrating our data with everybody else's, and we are working on generating and creating these web interfaces. Obviously, the Allen Institute is, has been ahead of the game, but I like to. I'm sorry I cannot be there today because it would be nice to really discuss further how to merge our data, and especially because we have seems like we have different brain regions. We done the hippocampus, you have superior temporal gyrus and prefrontal cortex, so that would be amazing. Yeah.
Thank you. So thanks for for the very good session and very good discussion. This ends the virtual presentation for today. For everyone here in person, please join us next door for meeting the founder session. Thank you, everyone.